algae section of the chapter, that is the second unit. So we are studying different types of types and their life history. In the algae we have already studied the general characteristics, the classification of algae, then also the reproduction in algae. So far we have studied about nostoc, chlamydomonas, algae. Now next, another important example under algae, that is the green algae, it is Oogonium. So Oogonium is a algae which is found in fresh water. It is sometimes found submerged inside water, attached to some hard substratum like rocks or any other solid objects inside water. And it is also found growing on the other organisms. It is found growing as an epiphyte on the surface of other algae which are larger. For example, on the red algae or brown algae which are large in size, they grow to a gigantic in size. So on the surface of their body, that is algae, Oogonium grows as an epiphyte but not a parasite. So just for its support it will grow there, and it is having chloroplast, that is chlorophyll. It is having the capacity of photosynthesis. Then sometimes it is found growing on the leaves and stem of submerged angiosperms, aquatic plants. So when it is found even on the roots also which are floating there, so on the roots and stem and leaves of the plants, Oogonium is found growing on the surface. This is about the occurrence of Oogonium. Now if you come to the nature of the body, plant body, algae, so it is a thallus definitely. So usually algae are all thallus. They don't have well defined root, stem and leaves. So here the thallus of the Oogonium is filamentous. So that means it is made up of elongated cylindrical cells which are found in a row, and they are unbranched. That is also important character. So the cells are filamentous, unbranched. They are attached, the filaments are attached to the substratum. Now suppose this is a substratum, so it may be a rock, it may be any other hard surface. So this filaments are growing like this. Suppose this is a cell, this is another cell, and this is another cell. So these are the cells of the Oogonium. The cell which is attached to the substratum, it is somewhat different, and this is called as the holdfast. So the basal cell, which is elongated and broad at the base. Now this broad at the base, it is having some projections like some finger-like projections. So they help to have a proper grasp to the object and helps to fix the filament properly to the substratum. So that is the holdfast cell, which is usually an empty cell, and then it helps in fixation of the filament. Then, so that is about the thallus. So the thallus is filamentous, and it is made up of unbranched, elongated cylindrical cells, and the holdfast, the basal cell, helps in fixation to the substratum. Now if you come to the cell structure of a single cell, so a single cell, suppose this is a cell, so an elongated cell like this, and the cell wall is made up of cellulose and pectin. So cellulose is on the inner side and pectin is on the outer. So usually there is cellulose on the inner side and pectin on the outer side. So the cell wall is made up of cellulose and pectin, and sometimes the third layer is there on the outer side, which is made up of chitin. So on the inner side there is cellulose, and on the middle there is pectin, and on the outer there is chitin, that is on the surface. So the surface investment is chitin, which is considered as the third layer. The first layer cellulose, the second layer pectin, the third layer chitin. So these are the composition of the cell wall. And coming to the other parts of the cell. Now this cell, it is having the cytoplasm and the nucleus. Now the very important thing is the chloroplast is somewhat different here. So the chloroplast is branched like this. So this chloroplast is reticulate in the sense. So this reticulate chloroplast is present from one end of the cell to the other end of the cell. It is extending from one end to the other end. Reticulate chloroplast. Branched. And these branches are somewhat broad or somewhat narrow than the branches. So this branched chloroplast is present, and again it is somewhat cylindrical in shape. So in that cell the chloroplast is in the outer part of the cell. That means it is hollow and it is extending from one end of the cell to the other end of the cell. That is the nature of the chloroplast. Then if you come to the other parts of the chloroplast, now in the chloroplast you will find some proteinaceous bodies. So these proteinaceous bodies are called as pyrenoids. So there are many pyrenoids in the chloroplast. So the pyrenoids are protein bodies and they are present in the chloroplast region. So the chloroplast, it contains as usual the green pigment chlorophyll. Then there is a nucleus, single nucleus, and the cytoplasm contains all the cell organelles like mitochondria, Golgi complex, endoplasmic reticulum, ribosomes and all that. That is about the structure of the single cell of an Oogonium. So mainly the cell wall is the cellulose, pectin, and the outer is chitinous. And inside the cell, as usual, there is cell membrane again. So inside the cell wall there is the plasma membrane, and within the plasma membrane we find the nucleus, cytoplasm, cell organelles, and the reticulate chloroplast, which is highly branched. So the reticulate chloroplast, which is branched into many broad and narrow mesh-like structures, long finger-like structures. So they extend from one end of the cell to the other end. The chloroplast is hollow and it is cylindrical in shape. It contains chlorophyll, and there are pyrenoids in the chloroplast. So pyrenoids are some proteinaceous bodies in the chloroplast. Then if you come to the other parts of the cell, so this cell contains the cellulose, pectin and chitin, which are parallelly arranged in fibrillar manner. Then if you come to the reproduction in Oogonial, so the reproduction takes place by asexual and sexual methods. So the asexual method of reproduction and sexual both. So first we shall see the asexual type of reproduction. So asexual type of reproduction in Oogonium takes place by the vegetative method and as well as sporulation. So vegetative method and sporulation is the two methods of asexual reproduction. So vegetative methods, it is a simple method of fragmentation which we have already studied in the nostoc. So the nostoc filament, so same as the Oogonium filament, which is made up of elongated cells. So the nostoc filament, which is where the filaments break, here also the Oogonium filament, when it breaks accidentally into segments or which are called as fragments, each fragment is capable of developing into a new filament by simple cell division. The cells will divide by cell division and the number of the cells increase in the filament, and this filament, when it comes in contact with a hard object, a solid object, so the basal cell will become the fold fast and that attaches to the this substratum. That is in case of vegetative type of reproduction. Now if you come to the sporulation, production of the spores. So this spore production is by the help of zoospores formation. So zoospores are formed in the cells. So the zoospores, how they are formed in the filament. Now suppose this is a filament, this is a cell. So the zoospores are formed by the, suppose any cell may form a zoospore. Now this zoospore, here there is a sufficient storage of the reserve food material, and after the storage of the reserve food material, here what happens, a single cell will produce a single zoospore. So this cell, when it forms here, here you will find this chloroplast like this, and this is the nucleus, and when in the next stage what happens, the cell membrane separates from the cell wall, and at one end a colorless area develops like this. So a clear area develops at one end. So this is the nucleus and this is the zoospore. So this colorless area is formed as an hyaline area, it is called as, and at this end there are numerous granules arranged inside the cell. These are actually blepharoplasts, granules means. So blepharoplasts, as we know, they give rise to the flagella. Similarly, here also there are many blepharoplasts which gives rise to the flagella. So these flagella are produced on the outer side of this cell inside. Now this is actually the zoospore. So the zoospore which is formed in the cell, so that zoospore is formed by the accumulation, by the storage of the reserve food material. So that reserve food material is stored, and then the cell membrane withdraws from the cell wall and forms a clear zone at one end. where certain granules accumulate or become arranged. So these granules are actually the blepharoplasts. They develop into blepharoplasts which produce the flagella. So like that there is a ring of flagella produced. Sometimes there may be two rings, one or two rings of flagella are formed at the one end of the this structure, and by the rupture of the cell wall, so this zoospore which gets separated. So this is the chloroplast region, and this is a clear area which is having the flagella. So this flagella, a ring of flagella which are produced. So this is the zoospore which is produced from the single cell. So this a single cell produces one zoospores. So that zoospores which are released from the cell, so these swim in the water by the help of flagella, and when they come in contact with any hard substratum, they attach to this hard substratum by the colorless area. So the colorless area attaches to the substratum. So then this zoospore will divide by mitosis. So this part of the zoospore becomes the holdfast, and the other part it develops into the filament. So like this, the zoospores, they are developed from the single cell, and then they are having this type of flagellation. So this type of flagellation is having a particular word called as stephanocont. So stephanocont means the arrangement of the flagella in a ring. So one or two rings of flagella are present at one end, that is because of the granules which are present. So this end attaches to the substratum and it develops into a new filament. That is how the zoospores, they develop into the new filament. Then if you come to the another type of sporulation, so that is by the help of akinets. Again in nostoc we come across such kind of akinets. So zoospores are usually formed during favorable seasons, and again the zoospores depend upon the carbon dioxide content in the water. So the presence of CO2 will affect the production of zoospores. So but when there is unfavorable conditions, so when the conditions are not suitable for the growth, and then some of the cells in the filament, they develop into akinets. So in the filament, certain cells, they become thick walled. The wall becomes very thick, and there is the storage of the food material. So these cells are called as akinets. So the akinets, they survive in extreme conditions, and in the coming favorable season or in the coming favorable condition, so they develop into a new filament by the attachment to the substratum. So the thick wall gets released, separated, and then the akinets, they become attached to the substratum, and they then divide to form a new filament. That is how the zoospores are, the akinets, they help in overcoming the unfavorable situations in the season. So but zoospores, they are produced usually in the favorable season. Then if you come to the sexual reproduction of this Oogonial. So in the sexual reproduction, so when we say that there is sexual reproduction, definitely it is the presence of male and female reproductive organs. So the sexual reproduction takes place in Oogonium. So there are two types of species in Oogonium. So macrandrous and nanandrous species, depending upon the nature of the sex organs. So macrandrous means they are the normal type of filaments. So the male reproductive organ, it is the Anthridium. The female reproductive organ, it is the Oogonium. The sexual reproduction is oogamous. So we have seen there are three types of sexual reproduction in algae: isogamous, anisogamous and oogamous. So here we find oogamous. So oogamous is the advanced type of sexual reproduction. So where the gametes are produced in well defined structures, that is male or female gamete, but in case of iso and anisogamous, the cells directly themselves they give rise to gametes, and in isogametes they are similar. We cannot differentiate the male and female gametes, but in isogamous there is in anisogamous there is the differentiation, but here there is oogamous type of reproduction. The male gametes are produced in Anthridium structure and the female gametes are produced in Oogonium structure. Now in macrandrous what happens? The Anthridium, this macrandrous and nanandrous are concerned with Anthridium. When the Anthridia are produced on normal filaments, it is macrandrous. When the Anthridium or Anthridium is produced on small specialized dwarf filaments, they are called as the nanandrous. So the word itself we can find out the meaning. Macro means large. So these in the large filaments, normal filaments. Andro means male. Macrandrous. So macrandrous means the Anthridium. Andro means male. In flower we refer as andrecium, that is in higher plants. So here Anthridium is a normal macro. When the Anthridium is small, nano means small. So they are produced in the short filaments, which are called as dwarf filaments. So these small dwarf filaments, they produce Anthridia, whereas the normal filaments produce Anthridia. So that is on the basis of the nature of the sex organs, particularly the Anthridia, the sexual reproduction or the type of the reproduction in Oogonium is macrandrous or nanandrous type of reproduction. So in macrandrous the filaments are normal. In the nanandrous the filaments are short, particularly the Anthridium is very short. Now what? But the production of gametes is same. Only the nature of the filaments they are different. So in the nanandrous the filaments are somewhat particular in nature. So these male and female reproductive organs, they are nothing but the cells of the filament. So in a normal filament cells, suppose in this is a normal cell. So any cell of the filament it produces. Then another important thing in the structure of the cell to tell you here is, now in some cells there are some ring-like structures on the upper side of the cell. Suppose if this is a cell, this is a ring-like structure seen. So these ring-like structures which are seen, they are called as apical cap. So that means that a ring-like structure is actually made up of hemicellulose, but hemicellulose is not present in the cell wall. We have already mentioned that the cell wall is made up of cellulose, pectin and chitin. Inner there is a cellulose, middle there is pectin and outer there is the chitinous layer. But in some cells we find a ring-like structure, sometimes 2 or three rings are present. So that is at one end of the cell they are present. So these rings are actually hemicellulose rings at one end of the cell. So such rings which are seen, they are called as the apical caps. So at the one end of the cell they appear like a cap like structure at the apex or the end of the cell. That is why they are called as apical caps. And such cells which are having apical caps, they are called as cap cells. When there is no apical cap, they are normal cells. So this cap cell means it is somewhat a developed cell. In the same way here, in the sexual reproduction, the cap cells, they involve in the production of Anthridium and the Oogonial. So in Macrandrus, the Anthridia are produced in normal filaments. In Nanandrus, the Anthridia are produced in specialized dwarf filaments. That is why Nanandrus. Now here again, the Anthridium and Oogonia, they may be produced on the same filament. That is monoecious, they are called as. So on a single filament, some cells they produce Anthridia and some cells they produce Oogonia. That is monoecious. And there are also Idogonium species where the Anthridia and Oogonia are produced on separate filaments, on different filaments. That is dioecious. So dioecious means the separation or the production of the Anthridia, the formation of the Anthridia and the Oogonia on separate filaments. So we come across both types of filaments in Idogonium. The Idogonium may be monoecious or dioecious. So even in Macrandrus, there may be monoecious type and there may be dioecious type. So now in the production of the Anthridia, if we come across again, so the Anthridia are produced in a well-developed cell, in any cell of the filament. Again here, the cell will store, there is accumulation of the reserve food material, and again the presence of certain environmental factors affect the sexual reproduction. For example, light, CO2, hydrogen ion concentration are very important. So they are the factors which are necessary for the reproduction, sexual reproduction. There should be proper amount of light, there should be proper content of the CO2 and also hydrogen ion concentration. So that means the acidity of that pH of that medium also will affect the process of sexual reproduction, and also the nitrogen deficiency is another factor which also affects the process of this reproduction. Now the Anthridium is any cell of the filament. It functions like an Anthridium. So here the cell will store food material and it will divide by mitosis. The two nuclei which are formed in the cell, each nucleus forms a cell and it will form a male gamete, or what are called as the sperms. So these sperms, they are similar to the zoospores, but zoospores are somewhat different. A sperm is having a ring of flagella. It is also having a colorless area like a zoospore. So these zoospores are having many flagella, but the sperms are having only few number of flagella, but not as many as zoospores. Sperms are also motile. Zoospores are also motile. They move freely in the water. Zoospores, when they are released, they move freely in the water, and when they come in contact with a hard or any solid object like a rock or like that, they become attached. Here the male gametes, the sperms, they are released by the rupture of this wall of the Idogonium. So the cell of the Idogonium gets ruptured and then it will release the sperms in the water. Now if the filament is monoecious, the oogonium is present on the same filament. But when the filament is dioecious, so the oogonium is present on the different filament. Suppose here, if this is the oogonium, if we take into consideration as the monoecious filament, so a single cell will produce the oogonium which functions. So the cell which produces the gametes, they are called as the gametangium or gametangia, they are called as. So the male gametangium is the anthridium, the female gametangium is the oogonium. So the male gametangium produces a pair of male gametes or sperms, and these sperms are also flagellated, they are motile, they swim in water and they move freely. Now next what happens, if you come to the oogonium, the female gametangium functions as the oogonial mother cell. So any cell of the filament, it is the female gametangium and it is called as the oogonial mother cell. This oogonial mother cell divides by mitotically to produce two cells. So here when the two cells are formed, one cell it forms the oogonium after a division of the oogonial mother cell. The other cell is called as the support cell, or it is also called as the suffoltory cell. Suffoltory cell or support cell, that is the name given to that cell. So it is just a cell which gives a support to that oogonium. So one cell functions as the oogonium, the other cell is a just support for that. So the oogonium differentiates into the female gamete, egg or ovum. So this egg or ovum, it is a female gamete. So again the cell membrane or the plasma membrane, it gets separated from the cell wall and then it forms a particular structure. At one end of the oogonium, there is a structure which is called as receptive spot. So this receptive spot is a region in the oogonium. So this receptive spot, and these oogonium or sperms, they are usually green in color, but zoospores are somewhat brownish red in color. So as the chlorophyll is present there, so usually they appear green in color. They are motile, that is zoospores and the male gametes or sperms. When the oogonium is formed, it means it is having a receptive spot. A single oogonium produces a single egg, but an anthridium produces a pair of sperms, remember. And in the asexual, there the single zoospore is produced by a single zoosporangium. So this cell is called as the zoosporangium, the cell which produces the zoospore. But in anthridium, two gametes are produced. But in oogonium, single egg or ovum is produced, and this oogonium is having a reserved food material and it is having a receptive spot. So when there is receptive spot formed, it means that the oogonium is well developed to receive the sperm. Now when the oogonium and the sperms or the anthridium are on the same filament, that is in monoecious, so the sperms enter in the same filament there. When they are in different filaments, the sperms will swim or they move towards the oogonium on a different filament. Now the oogonium, how the sperms they move towards the oogonium. Usually it is a general aspect actually, even in the higher organisms, whether it is an animal or other organisms. The oogonium as well as the sperms, they release a kind of chemical which diffuses in the surrounding medium. So this oogonium which releases the chemical into the surrounding medium, so that chemical will attract the sperms towards it. So as a result, the sperms will move towards the oogonium. But actually the sperms also will release a kind of hormone. So it is chemically a hormone, but generally we say that it is a chemical. So that chemical is diffused in the surrounding. So the chemicals of the two, that is the sperm and the ovum, they interact each other. As a result of interaction, if these two are belonging to the same species, the sperms will move towards the oogonium. But suppose if the sperms and oogonium are of different species, they will not, the sperms will not move towards the oogonium. So that means there will be no interaction between the chemical produced by the oogonium as well as that of the sperm. So the result of the interaction between the sperm and the oogonium means it brings about the movement of the sperm or the locomotion of the sperm towards the oogonium, as the sperms are motile due to the presence of flagella. The oogonium or the egg or the ovum is non-motile. It is present within the oogonium itself. But the sperms will come out from the male gametangia, from the Anthridium, and move freely in the surrounding medium. But the egg in the oogonium does not move out because it is non-motile. It does not possess any flagella. So thus one of the sperm will enter into the egg through the receptive spot. So the receptive spot is the region through which the sperms are received by the egg. And when the sperm enters into this receptive spot, so there the fertilization takes place in the oogonium. So if it is dioecious, the sperms are moving towards the oogonium on other filament. Suppose if it is monoecious, the sperms will move towards the oogonium on the same filament. They are all along the same filament. So an adjacent cell may be Anthridium and oogonium, or there may be other cells in between. So if it is adjacent cells, the movement of the sperms is very easy. So that means there is no long distance for the movement of the sperms. So but whatever it is the main cause is the chemical which is produced by the oogonium. That oogonium will release that chemical and make the sperms to move towards the egg cell or the ovum. And here in case of Oogonium, there is a particular region called as the receptive spot through which the one of the sperm will enter and bring about fertilization. So a diploid zygote is formed in this region. Now this diploid zygote which is formed, it becomes thick-walled. So the zygote becomes thick-walled. There are about 3 wall layers formed around the zygote, and this structure is called as zygospore. So the zygospore is a developed zygote. Zygote means it is as soon as the fusion of the sperm and this egg nucleus, it is the zygote, that is the diploid zygote. So the diploid zygote, soon it develops a thick wall of 3 layers for its protection, and it is called as a zygospore. So the zygospore, only under favorable conditions it will develop further. Otherwise it will remain for a period of rest. So it undergoes meiosis. So it divides, and one of this diploid oospore undergoes meiosis there. Suppose this is the zoospore which is formed. This is the zoospore which is having a thick cell wall around it. This is the zoospore. So the zygote was without any any extra layers there. So the zygospore divides by meiosis to form 4 nuclei. So these 4 nuclei are formed in this zygospore. So these 4 nuclei, they develop into the what are called as the meiospores. So the meiospores are actually having a flagella, just like the sperms and zoospores. So presence of many flagella is characteristic feature here. So here also we find many in case of the sperms. In the zoospores also we find many flagella. Similarly here also each forms a cell, they are called as the meiospores or meiospores, they are called as. So each meiospores is having again flagella there, just like the sperms. So there is a ring of flagella formed. So the 4 nuclei which are formed, they become differentiated into 4 unicellular structures. So these uninucleate structures, they develop into what are called as the meiospores or meiospores, they are called as. So the meiospores, they are released by the rupture of this wall of the zygospore. So the meiospores, they swim and they move towards the favorable conditions. They move towards the favorable places. For example, if the water is polluted, so they do not develop there. They remain motile and they move towards favorable conditions or favorable places, and then they become attached to this any hard substratum. So any hard substratum, suppose this is the zoospore, so these are the flagella which are present, and this is the cell, same as that of the zoospores. So these are meiospores. In case of asexual reproduction, we have just mentioned zoospores. They are also multiflagellate due to the presence of many granules inside. Here also they are having many flagella. So these meiospores, they attach to any hard substratum, and then the cell starts dividing, and then it will develop into a new filament. So that is how the sexual reproduction takes place in Macrandrus and Nanandrus. So in Nanandrus, what happens? The Anthridae are produced in short filaments, specialized filaments. So these short filaments, which are highly reduced, and they are somewhat dwarf in size, they are small in size, they are highly reduced filaments. So they are called as Nanandrus species or Nanandrus filaments, they are called as. So that is how Macrandrus and Nanandrus they develop into the Anthridium. They develop the Anthridium and Oogonium. The Anthridium is developed from the normal cell. Usually a cap cell develops into Anthridium. A cap cell is a well developed cell, a matured cell, and each functions like an Anthridium. The male gamtangium, a pair of sperms are developed, so it divides and 2 sperms are released from this cell, and the Anthridia, they developed, they are liberated by the rupture of this cell wall, and then they move towards the Oogonium, that is by the attraction of the chemical which is released by the Oogonium. So fertilization takes place in the Oogonium, which the sperm enters through the receptive spot and fertilizes with the egg, and this is the support cell or supportory cell, it is also called as. Then the zygote which is formed, it develops, it is actually released out of the filament, and then the zygote develops 3 thick layers, and then the zygospore is formed. The zygospore produces 4 meiospores by meiosis, and then each mesospore is released. They are flagellated, they are multiflagellated, and then they swim freely in the water, and then they get attached to the hard substratum and develop into a new filament. That is sexual reproduction in the Oogonium, which is of macrandrous and nanandrous species. So this is about the life history of Oogonium.